Cancer is typically treated using multiple strategies, and one of the most widely used methods is combined chemotherapy. One example is the CHOP regimen, which includes 4 drugs: cytoplasmoid, hydroxydoropinic, oncovin, and prednisone. Each drug targets cancer in a different way. Cytoplasmide and hydroxydoropinic work by damaging DNA, while prednisone helps suppress inflammation and immune responses. Oncovin, which is one of the focuses of today's discussion, prevents cancer cells from dividing by targeting a specific protein involved in cell division called tubulin. Rather than directly damaging DNA, oncovin interferes with the machinery the cell needs to successfully complete mitosis. This unique approach is vital in halting the rapid spread of cancer cells. Tubulin is a protein that plays a key role in the structural framework of cells, particularly during cell division. It assembles into long, hollow tubes known as microtubules, which form part of the mitotic spindle. The spindle is responsible for organizing and moving chromosomes during mitosis, ensuring that each new cell receives an exact copy of the genetic material. When a cell prepares to divide, microtubules grow and reach out from the centrosomes to attach to each chromosomes, aligning them in the center of the cell and then pulling them apart to opposite sides. Without the proper formation and function of these microtubules, the chromosomes cannot be disrupted evenly, and the cell cannot divide properly. In this way, tubulin is essential to life and especially important to cells that divide frequently, like cancer cells. Oncovin, also known by its genetic name vinctine, disrupts each cell division by directly targeting tubulin. It binds to tubulin molecules in the cell and prevents them from assembling into microtubules. Because microtubules are required to form the mitotic spindle, this interference effectively shuts down the cell's ability to divide. The cell becomes arrested in a stage of mitosis called metaphase, where chromosomes are lined up but cannot be separated. When the cell cannot proceed past this point, it eventually triggers a self-destruct process known as apoptosis. This mechanism is especially effective against cancer cells, which are constantly trying to divide and multiply. By targeting a structure they rely on so heavy, oncovin helps to stop their growth without directly damaging DNA, making it a powerful component of the CHOP treatment plan. In conclusion, oncovin is an important chemotherapy drug that works by targeting tubulin, a protein necessary for cell division. Tubulin forms microtubules, which are essential for moving chromosomes during mitosis. By binding to tubulin and preventing microtubulin formation, oncovin effectively halts the division of cancer cells, leading to their eventually death. This makes oncovin a vital tool in cancer treatment regimens, especially for cancers made up of rapidly dividing cells. Unlike some other drugs that damage genetic material, oncovin stops the machinery of cell division itself, adding a unique and effective method to the fight against cancer.